coming back to today's event. Today is our 24th webinar of the series. We welcome all the viewers who have joined us from around the world. After attending today's talk, you can apply for a certificate of participation via the feedback link and password. We have already emailed the feedback certificate application link to the registered participants. The password will be provided multiple times in the YouTube chat box during the Q&A and interview session, which occurs after the lecture. Submission of the feedback form is mandatory to receive a certificate. When you fill out the feedback form, please mention the full name of your institute and the full institute address. You can collect your participation certificates after 2 days from our website. Bioingene does not send certificates through email. Please visit our website to register for upcoming webinars. We can connect with Bioingene via Facebook, YouTube, Twitter, and LinkedIn. I now request Jacinta to please introduce our speaker for today's webinar.
Thanks, Liberty. The topic for today's webinar is From Breeding to Genome Editing, Long-Term Strategies to Contain Plant Diseases by Ricardo Oliva. Dr. Ricardo Oliver is a senior scientist at the International Rice Research Institute, or IRRI, in Los Banos, the Philippines. He is the cluster leader responsible for 30+ staff, including scientists, postdocs, national staff, and scholars. He has over 10 years of research and development experience in staple crops in Asia, Africa, and Latin America. He is the recipient of research funding from key donors such as the Bill and Melinda Gates Foundation, the Biotechnology and Biological Sciences Research Council, BBSRC, and the United States Agency for International Development, USAID, among many others. He has coordinated scientific networks, including public and private enterprises. He has a strong publication record, developed online tools, as well as several patent applications. He has published in journals, including Nature Biotechnology, PNAS, Frontiers in Plant Sciences, The Plant Cell, Molecular Plant Microbe Interactions, Crop Research, Frontiers in Microbiology, the Plant Pathology, the Plant Journal, just to name a few. He has also published many book chapters and other publications. He has always been engaged in collaborative research with renowned research institutes from all around the world. We are honored to have him with us today. I now request Dr. Oliver to please share your slides and you can begin your presentation. Thank you.
Yes. Okay, sorry. Okay, you hear me?
Yes, yes, loud and clear.
Thank you. I want to thank all the people that is now in the broadcast for attending this presentation, and of course the group. You guys are doing a fantastic job. You know, it's like a very nice way to democratize science, and that's have a big value for all of us, and also in this moment of our lives. Yeah, thank you. You're awesome.
Thanks.
Anyway, so today I wanted to talk about the strategies to contain diseases. And when I say strategies, what we intend is durability, long-term, and efficiency. Because if you notice, diseases are still there, and we have been through many processes of trying to develop these strategies for the last 60 years, and we still have the same diseases, just hitting a little bit worse than before. So there's something that we are doing wrong, definitely. Okay, so now it's blocked, sorry. Okay, so common diseases that are in the news normally, you can see this banana disease in Colombia, the Fusarium oxysporum reaching Colombia for the first time, just setting a lot of alarms in the region because of the production. No, sorry, I have to. Looks like it's blocked. Okay, I have to go this way. In Bangladesh as well, wheat blast just hit in 2017 and set the alarms. In Europe, we have a lot of farmers in the olive industry are really suffering. And these are outbreaks that hit a global perspective. However, the outbreaks that we are looking for are the small ones, the ones that are building up on something bigger. And those are the ones that hit most of the farmers. Imagine at IRRI, what we are doing is trying to work with smallholder farmers. Around 164 million of farmers are out there in Asia. So if you are in that group or in that category, it's highly likely that you have or are close to the poverty line in most of those cases. So any event of crop failure or peak in the food crisis or in the food supply really affect you and affect the household. So those are the group of people that we are intent to help, and those people suffer small effects on the diseases at low scale. Just to give you an idea on the economic impact, this is a publication that was out in 2019, and this is a survey of a big survey on different crops, 5 main crops in different regions of the world. And if you have see here in the world, they measure the yield loss due to pests and diseases. And rice is actually the most affected. They calculated 30% of yield loss in rice. This is a cumulative. So that means that all the pests and all the diseases that are out there every year are taking 30% of the total production. So you can imagine that amount of income and amount of resources. The second point is also the environmental cost. For the environment, the farmers and the environment suffer a lot. You can see here a typical picture of farmers spraying chemicals. And here you see a comet test from a human cell that has been exposed to nothing, and a human cell that has been exposed to a chemical that is common in the agriculture. So that just measured the DNA damage. So definitely there is a component that human cost there. And if we want to understand the pathogen, we can just classify these into 2 main features. And those are the features that we need to attack or try to make solutions. So the first problem is that pathogen change. It just matter, is a fact, biological fact, right? Pathogens have the smaller genomes, are smaller, and can replicate. The life cycle is small. So they can accumulate mutation, and then they can evolve faster than the host, for instance, the plant. So if you have a mutation in a pathogen and make this pathogen virulent, this will accumulate during the seasons until the outbreak comes. This is the point when we see the disease, we go there, collect samples, try to work what is the relevance for those strains, try to understand what resistant gene we can use, and then we come back to the farmer after several years probably. But by then, the pathogen have changed already, so the cycle is never broken. So what we want actually is to try to detect these virulent forms very early in the process, right? So to do that, we need a lot of information on the genomics. We need a lot of information on the genetic component, the genes that are doing a part of the virulence. And based on that, what we need is to apply a small test or very reduced effort to detect those in the field. try to make interventions early on in the process to break the cycle as fast as possible. So if we can detect the pathogen early, we can act early and break the cycle. So that's the first thing. The second problem is that pathogens are not static, right? Pathogens do not have passport. They can freely roam around across the regions. However, the countries, the breeding programs, the National Extension Service, they do have passports, they do have borders. So any intervention that they take is local intervention. And you can see here in this test, in this map, Thailand, Cambodia, and Vietnam all have the same pathogen with a very similar virulent spectrum, right? You can see the photos here in Cambodia or in Vietnam. However, the interventions of these three countries are different. So definitely we need tools that help us visualize the problem and take decisions together, right? So that's the key point, to make interventions that have a regional context. Okay, before moving forward, I want to touch a little bit about cellular level. Why we say this is a co-evolving process between the plant and the microbe. And there's a lot of literature out there, so tons of papers. But this is what we know so far. So this is a very simplified way. Sorry if I have too simplified, but what we know so far is that either bacteria, fungi, or oomycete, or even nematodes approach the plants using effectors. The effectors are proteins that are secreted. In the first phase, what they are doing is simply help the pathogen to reach the cell, right? But in the first level of defense, the plants evolve these membrane receptors that the main point is to detect effectors and trigger a defense response in the plant. We call PTI. This defense response is the reason why all the plants are green. It will take out all 99.999% of the pathogens and keep the plant safe, okay? So, but there is some pathogens that adapt to this process, and they develop or evolve effectors that can go inside the plant cell through the type 3 secretion system in bacteria, or the haustorium is a structure that the fungus or oomycete use to invade the plant. So these effectors are basically plant proteins now, and the main target is to block PTI and allow the infection. Of course, they deregulate many of the processes, and that's kind of a subject of big research at the moment. In the third level of evolution, plant evolved the resistant genes that we call. There are NBSLRR type of receptors that detect the effector genes in the plant or the effector proteins and trigger a second type of resistance that we call effector trigger immunity. And this is ends up in cellular death. So basically the plant is a suicide mechanism, so the plant can stop the infection. And of course, pathogens evolve and kind of try to overcome the plant by modulating these effectors, meaning mutating the effector, changing the effector structure, or even deleting the effector. So this process is going on and on in all the host-plant interaction system that we know so far. There's some variations, and definitely there's many groups studying those variations, but this is the general concept. Okay, however, apart from immunity, we have another mechanism that is being studied recently that we call, or I call, starvation. And it's basically the plant generating mutations that block access to nutrients. Okay, the main purpose is that the pathogen, either fungal, bacteria, or nematode, got no access to the nutrients, sugar transporters, for instance, or sugar sources. Here is a little bit more of detail of this process, in which, if this is the apoplast and this is the cytosol, I don't know if you see my pointer. You see now, I guess. So infections of bacteria in the plant, what they are doing is inducing sucrose in particular, to be exported to the apoplast part for feeding purposes. In case of fungal, what they're doing is inducing glucose or fructose inside through transporters, of course, to feed the Haustorian structure here. So this process is very, very common and is being studied right now. And the reason why we are interested in this is because if we try to block this process of nutrient acquisition, it's highly likely that the pathogen cannot adapt to another source of sugars, right? So unless, or in comparison with effector genes or in immunity, pathogen is very good adapting to immunity processes of the plant. However, if you remove the source of nutrients, pathogen will have a lot of trouble to adapting to a lack of sugars, or try to utilize a different sugar. So that process probably is more long-lasting than the immunity per se. So in general terms, what we want is combine these two processes, either immunity and starvation process or starvation mechanism on the plant. Okay. So what you see now is somehow our equation, a working strategy in which we have a toolbox of resistance. So genes that we clone, that we create, do genome editing, or genes that we introgress from wild material. You have pathogen surveillance or pathogen understanding, pathogen monitoring. So what are the populations? What sort of races do we have? What sort of virulence factor do we have? And of course, the environment is important. So whenever we want to deploy any gene, we need to take in account the pathogen population and the environment. And every single season or every single breeding season is an opportunity to take a decision, right? So we are applying this sort of decision making to guide our breeding programs, at least at IRRI, and we try to do this with other partners as well. So try to make this like a rule of decision making every season, every breeding block. I will explain how it works and how we want this to work. And let's start from the resistant part. Okay. Today I will talk about not breeding per se, but more on genome editing part, which for us is a new breeding technology. I will focus first on bacterial blight. Is a disease that we have been studying for a long time now. It's caused by Xanthomonas oryzae, Pathovar oryzae. It's a bacteria, gram-positive bacteria. And Xanthomonas is a vascular pathogen. So get in the plant through hydatodes or small wounds, and it moved through the vascular system. And what it does basically block the circulation of the leaf, and the leaf die. And this lack of photosynthetic tissue generate yield loss. So far, we have been controlling these bacteria using XA genes. So our normal genes that we put in rice, but what gene goes where and for how long, and what is the durability of that gene, that's a matter of discussion, and it's something that we are working on a lot, or to develop new genes that we know are more durable. So to give you an explanation what's happening at cellular level, imagine that this is the cell and this is the bacteria. So bacteria carry these effectors that are called TAL. And TAL are transcription activator-like proteins. So these are big proteins, actually. Each genome have around 18 to 22 TAL effectors. As a transcription factor, they have a nuclear localized signal and activation domain but also at DNA binding domain. So this DNA binding domain is built on repeats. Each of the repeats, as you see here, recognize one nucleotide in the promoter region of a gene, right? So it's a very gene-specific or sequence-specific activation. So what happened is that the TAL effector is being secreted inside the cell, go to the nucleus, recognize this site or this sequence, and activate the gene. Now we know that this TAL effector activate genes that are called sweets. These are sugar transporters, and basically what they are doing is export sugar outside to the apoplast. So as I mentioned a couple of slides before, this sugar is utilized by the bacteria. There is a lot of replication, and the bacteria continue with the infection. So it's very, very smart way to approach the cell, and it's subject of a lot of research because of the delicacy and the specificity of this process. Okay, this has been shown several times. This is one of the papers we have been collaborating with. This is lines that have a reporter gene, gas reported in suite 11, for instance. And you can see the induction of this gene in the vascular tissue. Probably the cut here is better than you can see the cells start to induce the suite 11 when are inoculated with the bacteria. Here you can see the same, but in a time course experiment, 1 day to 5 days post inoculation. You can see here in this part, some cells start to induce the gene at 3 days, but at 5 days is already highly induced. So the bacteria have enough sugar to continue the infection. We also know that some individuals are able to activate particular sweet genes. For instance, this is a strain that is called PXO71 is able to induce sweet 11, as we see here in these transgenic lines, but not sweet 13 or sweet 14. However, this strain that is called PXO86 can induce sweet 14, however, is not able to induce sweet 13 and sweet 11, and so on. So we know that these 3 genes are important, and we also know that not all the bacteria have all the genes. So the repertoire of TAL effectors are distributed in the population. But together, like a conglomerate, they are able to induce sweet 11, sweet 13, and sweet 14. And this topic is, again, a very big topic of research about the sweet genes in general and how they facilitate the infection, not only in rice, but in other crops. And you will see some papers going out there on the function of sweets into how they facilitate the infection process. So our working theory, again, we have 5 major TAL effectors from the bacteria side, and we have 3 sweet genes. So what we want to do is to block or to change the EB site in a sense that we block the TAL effector binding, and this binding will block the expression of sweets in the plant, either 11, 13, or 14, or all together at the same time. Remember one thing, that what we want is just touch or block the recognition site, but not messing up with the promoter region. Because if we touch the promoter, then the gene cannot be induced normally, and then there is a trade-off for the plant. So we need to be very careful about that. So our hypothesis is that if we block this process, we may find a way to block the whole sugar or sucrose transportation towards the pathogen infection. So this is what we did, actually. This is